these data sets that can improve the accuracy of the reconstruct evolutionary trees. Because whenever you take one character or two characters or smaller number of trees, that cannot give that result. We reconstruct the evolutionary trees by taking or by addressing the incongruences between different type of data. So, whether morphologists we are going to vanish? No way. But training is important. As they mentioned that the Indian students, they are very good as far as statistics is concerned, regards for a state that they are not trained in new techniques. It is very, very important that we should also learn new things so that if they want to do any taxonomic review or any sort of molecular work, their basic training is important, and that's why we have already organized other programs, and as I mentioned yesterday also that 17 years back, Dipti came all the way from, they were in Vienna, Austria, also Samuel and colleague, they all came to India to train our young taxonomists. And many of them, I think some of the senior botanists sitting here, they also were there present in that training program. So this is very, very important that morphologists should come forward, should not hesitate to embrace the new techniques, molecular taxonomy, DNA barcoding. We should come forward, and then we should not avoid the molecular techniques, these sequences, phylogenetic analysis, and so on. Otherwise, what will happen? Shrinking or even vanishing forever. So, if we will not evolve, then we will vanish like dinosaurs. So, the molecular techniques have transformed the phylogenetics, integrating morphological data. It will, to understand the evolution processes, we have to integrate both molecular and morphological data, and go further. Both genomic technologies and morphological analysis techniques, balanced approach that will both data sets will be crucial for reconstruction. So the message is we should take into morphological and molecular data for our studies. Why I am emphasizing that young generation, they want to publish their papers in high impact journals or international journals, but they are not going to publish your like species you may publish, but other data, if you are doing only floristic work, that as you already know, you have been publishing, it will not be published. So integrate your work, collaborate. As Dr. Dipti mentioned, that you have to collaborate with Indian scientists or the scientists abroad to do good research. And we have been doing this. We have collaborated with RSUC and many others, Switzerland, and so many distinguished taxonomies in the world. So the young students especially, you have to learn new techniques and do good research. All the best. Thank you.
Good morning to all of you. Very good morning to Sir, Professor A.K. Pandey. The paper you have put say representative is the most like in music, and the way you have given all the informations like morphology and comparative data, phylogeny and phylogenetic tree, fossil data, evolutionary biology, DNA barcode, integrating morphological data, and essential and relevance of Ebola. And these are very important for the young scientists like our generation. So I would like to congratulate all the members present here for hearing this paper, and I am very much thankful to you, co-chair, and I have listened your paper with a very attentively with these viewers. I offer my sincere thanks and gratitude to the organizers for giving me an opportunity to co-chair the session. Thank you, sir.
Thank you so much, sir. I also believe that we have to blend the old with the new in order to get ourselves heard. So now I will request our session chairperson and session co-chairperson to kindly felicitate Professor Pant. Yeah. We are just waiting for our next speaker. He will be joining us from the Czech Republic, and please allow us some time before, like he's there already, yes. So we will just start once the ball is set rolling. And actually the next lecture was of Dr. S.S. Das from BSI, Botanical Survey of India, Kolkata. So since our foreign speaker is waiting, and we will first finish this, and then sir has kindly agreed to present after this one. So thank you, sir. Thank you for this understanding.
Hello, Peter.
Pretty good to see you. Great.
Thank
Good
you.
morning.
You may continue now. Okay.
Hello, sir.
Okay, so I will share my screen, right?
Yeah, you can do that, sir. So I'll just introduce you a little bit for other listeners. So we all welcome Professor Peter Sakal. He is currently working as a professor in the Department of Botany, Faculty of Science, Palaki University in the Czech Republic. So he's joining us from there. Welcome, sir, to Assam University culture, although in a virtual mode. So I hope you will like your presence here virtually. So Professor Peter Smikal has specialized in legume domestication using pea, lentil, and chickpea, with focus on seed dormancy and pod dehiscence as key domestication traits. He is also working on the analysis and use of genetic diversity, particularly in the wild relatives of legumes. He has a huge number of publications, about 125 scientific publications, and he is doing active research right now. So I heartily welcome Professor Smikal, and I hand over the dais to him for his speech. Thank you, sir. Yeah, just one announcement. Please keep your mobiles switched off. Okay? Please attend this. This is a great opportunity that you all are getting. I'm particularly telling this to the students and the scholars. Switch off your phones. Okay? Please listen attentively. Thank you.
Okay, so once again, thank you very much and good morning, everyone. Just to start the screen, is it okay?
Yes, sir. Yes, so we can see your screen clearly. No problem.
Perfect. Okay, good. Good. So I'm really glad to be invited. I particularly thank to Dr. Debjuti Bhattacharya, which actually invited me. He has been some years back visiting my lab. So that's great opportunity. I definitely would like to join you, but that's impossible for a moment. So as you can see the title and as it was introduced, I will share with you some, let's say, view on the legume crop phylogeny and its utility for the, let's say, science and breeding as well. Just to introduce where I come from. So our university, as you can see, is quite old. Well, we celebrated a year back 450 years anniversary. It's a classical university, so it includes all faculties. And I think the student, in fact, was Georg Mendel, which before he went to Brno, which is a famous place for him, but he really studied in our university for 3 years as illustrated. So we are old university town with 25,000 students currently. My interest in my group is just illustrated in this snapshot. So we are not big group, it's small group, but we are interested mostly in a domestication, as mentioned. And in particular, we use several models, P primarily, to understand what happened between the wild progenitor and the current crop. Specifically, it includes, or we focus on the understanding of the seed dormancy, which is a physical type of dormancy because of the impermeability of the seed coat, and also partly about shattering or dehiscence. And we studied from various angles, so this starts with anatomy, but also genomic and metabolomic profiling. Just to let you know what we are really interested in. And the mentioned legumes. So legumes are quite known. It's very large plant family. Currently, it includes about 850 legumes genera with about 23,000 species. And as it is illustrated, we find them basically from tropics to Arctic, from Seattle to, and they include herbs, but also trees. What is quite, for use, it has various uses. I've got one of the main is because there's protein, rich source of protein. And what is also important, they typically, most of them are able to fix in atmospheric nitrogen, so they contribute to the balance of the winds. If you look at subfamilies. Actually, the most important in terms of the utility for humanity is the Papillonoideae, which is highlighted in yellow. The subclade Hologalagina includes the wide range of the genera, like chickpea, latirus, lentil pea, and so on. So these are really important legume crops. The others are important for other things, including timber, and many of these are found in the tropics. Well, if you look at globally, what is important, the legumes really contribute about 1/3 of our diet rotate, which is definitely a large number, and this is really important. And I mentioned that if they fix the nitrogen. Yeah.
Yeah, excuse me, sir.
Is it okay?
It is okay, but your slides are not moving actually. It's stuck in the first slide. Maybe you put something about that. Yeah, now it is moving. Now it's moving. It was not moving till now.
So, but is it over the whole screen or not?
We can see your screen. We can just see that this is the economically exploited legumes. So your slides are shared, I guess. Are you in the slide share mode, sir?
Yeah, I do have the slide share.
Yeah. So
I had it just like presentation, so I was wondering why. But like I can do it in this way if it's okay.
Okay.
Well, mentioning the economically important legumes. So this is the portal which curate the legume data globally, I would say. It's include, of course, taxonomy. There are several, let's say, curators, and also the phylogeny part, which includes genomic as well. As it is illustrated here, currently there is nearly 800 genera and 20,000 species included. I'm moving to the part which I'm interested. So, as mentioned, the legumes. So the Papillonoideae, the yellow, is the richest. They include about 500 genera and 40,000 species, including those which I mentioned already, which means soybean, pea, chickpea, lentils, and so on. And this is basically phylogenetically the most derived part of the legumes. If we go far down into the phylogeny classification, so into the tribes, I highlighted in a box the Viziae, or currently it's more accepted to call it Fabae tribe, which includes the 3 species which I will be talking mostly of today. Right. So this is, yeah, I wonder how you can, do you see that like this? Is okay? Yeah. So this slides illustrate, it's, as you can see, it's from 2012, but nothing really changed since then. This is a molecular study of Fabae tribe, including wide range. There is like 400 species included. And as you can see, the branches, I'm not going too deep into the branches, but what is quite obvious, that if you see the Vitia or Latirus parts, they're like splitted. So basically they looks like a paraphyletic. And also, as you can see quite clearly, that lentil, lens, or pea, pisum, is just one small branch of this phylogenetic tree, which suggests that this species, actually, they shouldn't be recognized perhaps like a genera, rather than it should be included into the larger part, either Vitia or Latirus. And this is currently heavily debated how it should be treated. Because this should be either one single genera. They're distributed basically globally, except of tropics. and there is like 400 or 500 species depending on taxonomical classification. This is the doom into the one of the branch, and what I pointed out, this kind of boundary between the Latirus and Vicia, where exactly the Pisum P is sitting. So if I move, do you see this screen now, or is it moving?
Yes, it's moving. It's okay, sir. Perfect.
Okay. So, okay, I will also. So this is how I suggested to rename it. It's actually have been curated by colleagues in Kugan in UK. So this is the, I think, quite known portal, and they suggest, and they actually actively do it, that the Pisum should be called now the Latirus oleraceus, so back something which Lamarck suggested and classified back in 1779, and lentil should be classified like Vicia lens, and the third species which I come on the end, this is somehow how I came to taxonomical part, was for a long time very enigmatic. It's named after the Vavilov, quite famous ethnobotanist, but currently it falls into the Latirus formosus. So this is what will happen for many species, including into this Faber tribe. And this comes to the part which I'm, let's say, this is the reason why I'm interested in the phylogeny taxonomy. Let's come to the progenitors. So there is a global, let's say, interest to basically investigate crop wild relative. So the progenitor of our current crops. Why they are interesting? Of course, they are interesting from evolutionary perspective, because as any other organism or species, they have been shaped largely by environment. So the habitat where they have been adapted to. Of course, what happened afterwards, people started to cultivate or basically make maybe first a clearing. They have been brought into the new environment, and they have to adapt. So they have to adapt to the human activities, and of course the human have traveled across the globe, so they have been gradually also shaped by the climate and humans together. Currently, basically, we do have the, let's say, very artificial agriculture systems where, well, basically they are monoculture in many cases. But we are coming back to the land races, let's say the traditionally cultivated crops, because of the value adaptation that's becoming more and more important. To remind you, what we rely on, actually humanity, our kind of food source is very fragile, because if you look at this pyramid, there are like 380,000 described species of, let's say, higher plants. We do have some knowledge about 50,000 of them to be edible or, let's say, useful, but only 250 or so we do currently cultivate. But on the very tip of this pyramid is just 4 crops: wheat, rice, maize, soybean, which basically are feeding humanity. So we are kind of very fragile in terms of the any problems. Back to the legumes. Actually, within these domesticated crops, plants, legumes are very important because together with cereals or grasses or poaceae, there is a largest number of species which has been domesticated by human compared to others families. It's nothing to do with the size of the family, because, for example, the Orchideaceae, arguably, well, there are few species used, but basically not really many. So altogether, we do have knowledge about 2,500 crops from 160 families. Legumes, as well, you are sitting in a let's say in a most diverse part of the world. They have been distributed all over. So we do have, of course, like phaseolus coming from Central America. We do have peanut or groundnut from South America, or soybean from, let's say, East Asia. So they're distributed globally, and they have different histories. I'm particularly interested in pea and lentil and chickpea in some extent. And these three are very ancient crops. They have been, there are evidences from archaeology that they have been here and used by humans for 10,000 plus years, and they originate from the Middle East. What is shown in on the table on the right is also interesting part that, interestingly, human has taken typically like one species per genus, or a few more, which is just interesting why it happened only to some and not the others. So there are some interesting aspects to address by this. But it also suggests that there is a potential to eventually de novo, with the knowledge we have today, some other species perhaps, like sister species, or use them for a breeding, for a crosses. So I have been focusing, and I will show you some part of our studies in this region, which is, let's say, historically very interesting, but politically very complicated, as you know. I mean, today this is still again the area which is difficult to do anything, including Israel, Syria, Turkey, Iraq, Iran.
Your slide is not moving again. Would you please check it again, Peter? Hello?
Very good. So, okay, I stand down. From which part, sorry for that, I didn't know that it was disconnected. So from which part it was disconnected? Have you seen this? Yeah, from which slide you haven't heard it? How much back should I go? This one. Okay.
Lens. This graph, pyramid graph. Pyramid. Slide number 14. 1 4.
What was the last?
Yeah. Okay. Thank you so much.
That you haven't seen or you have seen?
You may explain it again.
Okay. So I will start with this. I will be in brief, just not to lose the time. So what I was saying here is that there are thousands of, let's say, 380,000 described species about of the higher plants. We do have some knowledge about 50,000 of them to be. But the pyramid, as you can see at the top, it includes like 200 or 300 species which has been used currently as a crop. But on the very tip, there are just 4, as named like wheat, rice, maize, soybean, which provide 3/4 of our food supply, which means the humanity is pretty fragile in terms of the food. And among that, I mentioned that the legumes. So of course, legumes are very important for the humanity as a feed, because together with cereals, with poace, they contribute the largest number of species which has been domesticated. If you look at globally, the legumes are Asian crops, so they come from different parts of the world, like common bean Phaseolus from Central America. We have Vigna, various Vigna coming from Africa or Asia, but we have also soybean coming from East Asia, so they are distributed globally. My interest is in pea, and we have records in our archaeology that the pea, lentil, and chickpea has been taken by humans, maybe first not cultivated, but let's say harvested, about 12 or 15,000 years ago already. What is shown in the table is also the point that typically, even so there are several other species within the genus, we typically have taken one or very few which has become crops. Which is interesting from evolutionary point of view, why it happened. Like in chickpea, for example, we have only one species was used, but there are about 44 of them which eventually could be still domesticated with our knowledge today. So there is a large potential. And I mentioned the Fertile Crescent, so the Mediterranean or the Middle East part, which is interesting but very difficult region politically nowadays and even in past. So this includes Israel, Syria, Turkey, Iraq, Iran. And that's where they come from. So this is how the typical habitat of pea, lentil, and chickpea looks like. I was fortunate that during years 2013 to 16, I have been able to survey this area together with colleagues from Australia and USA. And we have been able to collect many hundreds or thousands of samples, including seeds, to be deposited in the gene bank. And as a result of this survey, which is illustrated here, would become quite clear. If you look at the red dots, which represent the wild chickpea, it's very restricted geographically. It's quite narrow. If you look at the blue ones, which represent the progenitor of the lentil, so Lens orientalis, it's much wider, but even wider more is P. Pisum elatius, and this can be even more extended into the Mediterranean region. Because we do have the knowledge of the sites, so because of its accurate GPS positioning, and also we can derive the environmental parameters. So we can study also the environmental, basically, distribution. And that's also quite clear that the chickpea is rather restricted in terms of environment, which holds true also for the crop. So basically the crop inherited this potential from the wild. Compared to pea or lentil, which is much more flexible, basically it has more environmental range, and this is also something to be exploited in potentially in the breeding. This is a study actually where Dr. Debjuti Bhattacharya has been involved when he was visiting our lab. So we have been doing quite extensive survey of wild pea and looking at the genetic diversity in terms of the genome-wide assessment. It was thousands of gene samples at that time. So we look at the whole distribution range of pea, and we can cluster them into the genetic clusters based on the similarities. And also on the left bottom part, there is the classification based on the environment, which also that there is environments, like, of course, in Europe is much more rain compared to Middle East, which is dry and hot. And it has been look at the pea. There are two species currently recognized. The wild species, meaning Pisum elatus and Pisum fulvum, and you can see there is a overlap, but the fulvum is more specific. It has specific requirement of environment to which is adapted to. I always wanted to be able to compare between the species, which has partly some shared history, specifically pea, lentil, and chili, because as I illustrated, there's some overlap in distribution. So recently, and this is a paper published this year, we make a kind of global inventory of lentil, specifically looked for the progenitor, the Lens orientalis. Actually, this study illustrates also the power of genomic, because this has been by sequence completely. And actually, many of the samples which we obtained from various sources, which has been classified to be orientalis, it turned out genomically quite clear that they cannot be orientalis, that they are different species. Because if you have a well-established reference, then of course something which you might not be able to distinguish by morphology, it can be classified quite clearly based on the genomic part. So we make a kind of inventory. What it turned out also that similar to pea, there is an echogeography behind it. So, and it was interesting to compare if there is the genetic part and environmental part how related. I don't go too deep into that study because it will take whole lecture, but I should say there is no clear assignment. And one way to think about it, we think that human activities has been involved, because of course the seeds can travel by animals, they can travel also by human perhaps, or birds, and so even so the classification by environment is clear, the genetic part is not so in agreement. This is a study, actually there are not so many studies done in a wild collected species and samples of progenitors. So this is done by my group. We collected these individual samples from several populations in borders between Turkey and Syria and Iran. And we look at also the heterozygosity, which suggests the cross or open pollination ratios, which was always a question. So there are some indications that, of course, the progenitor is more open pollinated compared to the crop, which is self-pollinated quite strictly. And also we can see that there are populations which are quite distinct, but many are having more better relationship. We try also to model, because we know the locations, so if you can derive the environmental, which is today, you can see what is a suggested ecological niche, so to say the present distribution, which is shown in a green color. And the dots represent the sampling sites. So there are potential to find more samples, of course. But what is important is if we put the projected climate change scenario, which means less rain and more hot, you can immediately see that many of these sites will be affected. So they will have a problem because it will be too dry and eventually they can disappear. So this is kind of a conservation issue, of course. And this is going to happen to many of the wild plants, actually, with such a restricted distribution. What is here? So every dot represents one sample, and there are 1,000 of them. There are about 6,000 of them. The green ones are cultivated peas, including land races, and the color ones, like yellow or red, are the wild ones. You can see they are slightly shifted, so their distribution of diversity of the wild ones is different from the cultivated one, which suggests again that there is a potential to exploit it. Our interest is what happened. I mean, what was the progenitor? I mean, the closest together today progenitor to current pea crop. What is also interesting, we do have 2 domestication events in pea actually. It was debated for a long time, but it's quite clear. I think we show it very conclusively that Ethiopian pea, Pisum abyssinicum, so basically African pea, which is kind of strange where it comes from, which is on the left side, it's distinct from the pea crop. We know it genomically or karyologically because there is some chromosomal translocations, so there is also fertility barrier. But the question was, of course, where it comes from, where it is derived from. So you see, pea crop is still quite large. I mean, the diversity is broad, but there is one specific group of Pisum elativus which is closest, which is from specific geographical area. I'm coming now to the, let's say, the modern time. So we have been contributing, of course, the species are being sequenced, I mean the full genomes. Of course, it depends on the money, but many, because they have been useful, so currently there, of course, sequence many of the cultivated crops, but also become more important to sequence the progenitors. So, well, I mean, mostly the phylogenetic position didn't change much, which is good. I mean, that suggests that our classification based on the morphology or even some molecular aspect has been largely correct. But what it also provides, there is additional power, of course. We can see the block of the genes, so to see, we can see the arrangement of the chromosomes, which I think is illustrated here. So we can speculate, or let's say we have better idea about the evolutionary part of the genomes. So we can see which chromosomes have been changed across the evolutionary part. We also can clearly date basically for example, the polyploidy events, like in terms of soybean, for example, it's quite clear that there was one additional whole genome duplication about 10 million years back, which instantly separated soybean from the others. It's affecting, of course, many aspects of today. And so this is the power of genomic, of course. That can provide the resolution which we cannot have by other means. So in P, there are currently several genomes sequenced. There are several pan-genome, so-called pan-genome efforts to sequence many more, including the wild progenitors. And we start to have much better picture of the evolutionary part which led to domestication. We can estimate also some, let's say, the gene assignments and also the trajectories of timing. What it is good for? Actually, this is what I was doing in the past before I came back to university with one of my very good PhD students as a first author. We look at one specific gene which is providing the resistance to the virus, which is important. And we use, of course, all the knowledge of phylogeny, so to say, not just cultivated, that was not so important, but the progenitors. And that was interesting where these resistant alleles come from, and that there are also new ones. Actually, the Indian subcontinent, so your home, is actually the very interesting in terms of the alleles diversity. There is far more alleles in Indian subcontinent compared to original part, which is interesting because it suggests P is not from India, but it has been taken into the northern part of India some time ago, and maybe because of the pressure from environment, it has to evolve this resistance. So back to the progenitor. If we have all this data, we can do some modeling, which is maybe fancy or today. But if you look at the requirement of the P crop, which is on the left, you can see some red color, which illustrate the area which is under the stress, or basically not very suitable for cultivation. The green one should suggest the area which is, let's say, suitable for cultivation of the current crop. Compared to the wild progenitor, they have different distributions. Basically, many part of the Asia is still, there is a potential to utilize the genomes from wild progenitor. How to do it? I mean, this is something which we have been doing for over 10 years. There are many groups doing this worldwide, including, of course, colleagues in for example, in ICRISAT in India, in Hyderabad, for other species. So basically, you know, the domestication has introduced what is called a bottleneck. So basically, the diversity has been narrowed, and sometimes very substantially. For example, chickpea is dramatically reduced in diversity, and that was the reason why we have been searching for progenitors, because there is urgent need to have new alleles. So by doing the classical cross, nothing let's say new or spectacular, we try to reverse the bottleneck and reintroduce diversity again. Because this bottleneck actually, it's depicted, it's of course impact the DNA diversity. So if you look at the wild ancestor, and these dots represent the alleles, so of course the wild ancestor has many, the large diversity. Compared to the domesticated crop where the diversity was reduced, and in some cases, like for example genes which is responsible for so-called domestication trait, can be just one single allele. And of course it's not just one region, but it can take, let's say, some genes around it. including some resistances or substrates. So this is how it is affected. Back, let's say, okay, I've mentioned the gene banks, but this is kind of like what diversity is there. What is kind of unfortunate, despite effort, I would say, in my own experience, people which work in let's say, the breeding part, or taxonomical part, or systematics, are not well connected, because I recognize it's different purpose, different word, but it's a pity there is often lacking the connections, because they can benefit from each other quite a bit, because there is a knowledge, of course, which is needed. In terms of globally, we have, well, it's old numbers, but it didn't change that much. It's over the 1 million samples of legumes of various genera. But if you look at the percentage of or proportion of the wild species, it's very little, specifically for most of our crops, and that also includes the cereals, because most of the conservation effort was directed to the varieties or plant races, but very little of the progenitor. So there is an action to still to do some collecting when it's possible, and also actually to include something which was not so well done in the past. Meaning, of course, we didn't have a GPS positioning system until, let's say, 80s, and then many collections, including herbarium specimens, are collected well before that. Of course, there is a good description in many cases of the place where it was found, and it can be, let's say, digitalized, and we can derive the environmental information, which is useful for species distribution or for many aspects, including modeling. So there is a global effort. It's coordinated by the Global Diversity Trust, which is in Germany, the headquarters. And among them, there are also many legumes which are on focus, like pigeon pea, for example, very important crop for India, or grass pea, which is also very important, very much neglected, which is under, let's say, the survey. There is, let's say, this concept of gene pools, which is different from taxonomical perspective. What it means is essentially you looking by experience also the cross, which is like a reproduction barrier. So in P, for example, we do only one primary pool of progenitor, which is basically no problem to when you have fertile progeny. If you go a step farther, like secondary pool, which include the Ethiopian pea, which I mentioned, but also some full moon, there is a problem. There is a fertility issue, and in some cases the progeny is not completely fertile. As tertiary gene pool, which means you need some kind of help. Basically, after the cross, you need embryo or tissue culture or something to overcome the barrier. When I mentioned Vavilovia, actually this is me personally very special species, because it was a long time kind of enigmatic. Actually, I had a Royal Society fellowship some time ago, because it was basically unknown where it is occurring in nature. It was just in herbaria, in Edinburgh and others. We know that it's from Armenia, Turkey, Iran. We were lucky to find, and we started to make a kind of inventory. We took it also in a voucher, because as it is stated here in the title, there is a potential because it's perennial, it's a mountain, it's grown quite high, but it's very close to the pea. Actually, they're suggested to be able to cross it, which we very beautiful and named, of course, by Nikola Vavilov. So what the progenitor is good for? Well, of course, there are many, currently in many crops, we have many problems in various stresses. I'm not going to mention most, but this is argument why it's good to look at the progenitors. They can be a source of resistance to many of the abiotic and biotic stresses. For example, this is a study done by a colleague looking at the flowering of peas, the wild peas. And the pea is kind of a long day plant. So basically in a native environment, it's monitoring the length of the day, and when the day is getting longer, it's start to flower. But some, of course, specifically closer to the tropics or those in Ethiopia, they had to be basically day neutral, or they have to change the sensitivity to flower, to the photoperiod, otherwise they will not flower. So they have adopted the new alleles of specific genes which we know, which are required for monitoring of the photoperiod lengths. And to the final part, we do exploit this diversity now by doing so-called introgression. So basically, extensive crosses with the progenitor to be able to establish the series of the lines, which has, by recombination, just exchanged part of the chromosomes. We do grow them in a field. We know that there is a big variation, so of course they have the background from the wild, but also they have good background from the crop, which means that they can be useful already. And this is I think the way how we can exploit the knowledge from let's say some phylogenetic part, because we do need to know how distant they are, because this crossability of course depends on the distance. And the final part, or the final slide, is I mentioned on the beginning we are interested in a seed dormancy. So this is just illustration how we use the knowledge. We of course look at some specific pattern and genes which are involved in a seed coat, which is our main interest. And we have been exploiting the full range of diversity of the legume, specifically pea, but also in faba, and we have been going now to the other legumes in terms of the one specific gene, which seems to be important for the defense actually of the seeds in the soil. And to finalize, of course, I mentioned many parts, and there are many people which help us over the time. So this includes from many parts of the world, including some people from ICRISAT, and also from other places like Kew Garden and so on, and many funding agencies. So with that, okay, that's my contribution. I hope that I showed you a little bit of the utility of the phylogenetic or taxonomical knowledge in legumes, how it could be used for the breeding purposes. So I stop with that and I give it back to you.
Very good, Peter. Very good presentation. And, I will just say a few words and before I hand it over to you, sir. So, thank you so much, Peter, for your extremely informative and captivating presentation. It is indeed a great moment for us to hear someone from the same university as the father of genetics, Gregor Mendel, belonged to. You you are also working on peace.
Thank you very much. Sorry for connections.
You are also working on peace, incidentally, and you look a little bit like him as well. So I guess, yeah, you're working on peace, but of course on a very, very great depth and spread and reaching out globally. And I believe that this is exactly how science works, you know. You have to take up from your predecessors, from the great work that they have done, contribute from your side, and then hand it down to the future generation. So great work, Peter. Thank you again. And I hand it over to our session chairperson to please continue. Sir, please.
Okay, thank you very much once again.
Thank you, sir. Thank you.
Peter, she's the head of the Department of Ecology and Environmental Science in Assam University, Silchar. This madam.
Okay, thank you. Well, I hope that I will visit you one day.
Okay, sure. We also welcome you. Sir, session chair,
Yeah, I
sir.
will do it. I will try to do it.
Session chair, sir, Professor A.P. Das, retired professor of North Bengal University, Siliguri, India. And after that, he is now serving as a adjunct professor in Rajiv Gandhi University, Arunachal Pradesh. Please, sir.
Thank you.
I've gone through your beautiful lecture. So, entire audience was actually spellbound. Everyone was enjoying your lecture. So, thanks for delivering a beautiful lecture for the audience. Thank you.
Thank you very much. Thank you very much.
Any
Yeah.
question? One question. Any question? Okay. Okay, it's okay. Thank you. It is difficult to bring the people to the dais and so some physical.
Yeah, I know it's online, it's not the best. It's better to be together alive, I mean in person, so.
Okay, yeah, sure. So, hope to meet you someday. Okay.
Okay, bye for now and
Just a minute, Peter.
have a good time and conference.
Just a minute.
Yeah.
Our session co-chair, Dr. Tajuddin Khan, and now he has joined as principal in some college.
Namaskar, Professor Peter.
Great. Nice.
We are from
Nice to meet
7,000
you.
kilometers
Yeah.
from Assam University to your university, and we have listened to a very nice and in-depth lecture you have delivered to our members of IAAT and all delegates from different nook and corners of India and the abroad. So we are very delighted to have listened to your lecture, and it's a very informative lecture, and it is a very commendable one. So I offer my sincere thanks and congratulations, and hope to meet you in the future. Thank you, sir. Have a nice day.
Thank you
Thank you,
also.
sir.
Thank you.
Goodbye.
Okay, bye and enjoy the conference. Bye.
Yeah, this one carried out with a little less of technical glitches that we often experience. So now moving on with the technical session, we have the plenary lecture by Dr. Sudhanshu Sekhar Das. He is presently working as Scientist F, Additional Director, and the Head of Technical Division in Botanical Survey of India. He is also Adjunct Faculty Member, Department of Botany, Maharshi Dayanand University, Rohtak, Haryana. His expertise includes biodiversity assessment, revisionary studies, phytogeographic analysis, climate change assessment of plant diversity. He has a large number of publications, over 150 research papers, books, etc. And he is also the recipient of many prestigious awards. And I personally had known him till now through email. I guess it's a great opportunity to meet. Looking forward to your speech, sir. Thank you.
Good morning to everybody. Since morning we have listened to many talks regarding the morphotaxonomy. This is regarding to molecular biology, regarding to different disciplines from the Sri Lanka and also from the P genetics, phylogenetic research. I have been working here for last 20 years in the Northeast, since my first appointment in the Gangtok, and thanks to BSI, where it has given an opportunity for me. And I have seen the gradual degeneration of the floristic components of each of the ecosystem. Because I have analyzed, because basically I come from a background where I have studied for modeling for the different ecosystems and modeling for the, basically modeling of the how a forest ecosystem is remain sustainable. That's why from the day one, along with the taxonomic study of the different components and different what are they assigned to me, I have also marked the different ecosystems, how it is given. And I'm also very lucky that I'm being a part of the study of the National Mission for Himalayan Studies, where a large project was allotted to Professor Kohli and a group of scientists, academicians, where we have integrated the invasive alien species of the entire Indian Himalayan. And for this, here we talk about this today. I can't invest in the Northeast because I have personally worked in Sikkim, Mizoram, Tripura, and Arunachal Pradesh. And some of my friends, Dr. Singha and Dr. Lalbhai Chaudhary, who has worked in for the Sikkim sector and Manipur and Nagaland. And this, when we talk about the invasive alien issues, this is undoubtedly a cross-cutting issues, because the invasive alien species, as we know that, this is a non-native plant that causes a tremendous loss to the native species, and also the species which are the potentially the cause to harm to the environment, both economically and also to the as a for component in the ecosystem. That is if you see that, this is the one of the most and the second worst threat to the biodiversity next to the habitat destruction, because as we know that the habitat destruction is one of the most foremost threats to the ecosystem where what we're talking about. And it also the ecosystem, in ecosystem when they establish this, it produces substantial environmental loss and the economic damages, and which is irreparable. And the negative impact is this is multiplied by the climate change, recently by the climate change, by the pollutions and the habitat loss, and also the human-induced disturbances. And this is therefore the invasive species. This changes the community structure. This is the change the different kind of plant species is found in the ecosystem, and also the species composition of the native ecosystem indirectly or directly. Though we are not able to mark or not able to observe any of the change in the ecosystem, but slowly and steadily the invasive ecosystems spread into the virgin ecosystem, the native ecosystems, and it goes up to the place where generally the public or our human settlement has not been gone. And this is also the important to inherit effect of the change in the nutrient cycle and also the ecosystem functions, ecosystem relationship between the native species. Therefore, it is important to learn about how the species has been is going to invades the native ecosystems and also the different areas. And if you see the invasive hotspots, if you see, because according to the hotspots of the, because as we know that Western Ghats and other northeastern parts of the country and the Eastern Himalaya is part of the ecosystem, one of the hotspot. And if you see, if you correlate with this one, the 49% of the total geographical regions of India is predicted prone to the invasion moderately and high level of the climatic suitability. And if you see that the three states, what we are talking about today, the northeast from the Mizoram, Manipur and Nagaland, this is having 100% prone areas. And this has already been studied, and these are known as the high climatic suitability, classified for the hotspots of the very plant in India. Because the 19, if the studies revealed that 9 of the 17 ecosystems of the hotspots presently. And this is the not unknown to people. This is the policy, because if you see the different policies of the different legal literature, actually the biodiversity target behind 2011 also been targeted. This is the target says that by 2020 the invasive alien species pathway has to be identified and prioritized, and prioritized is to be controlled or eradicated, and measures are to manage pathway and prevent their induction in the established pattern. Similarly, when we talk about the sustainability development goals recently in the post 2020 KB, there is a Montreal protocol. So we have also this goal number 15. We have the target that introduce the measures to prevent the introduction and significantly reduce the impact of the invasive alien species on land and also ecosystems and also control. And if you see the national biodiversity strategy for the action plan, the important thing is this is talks about by 2025, because by 2025 and last as per the HC target we could not have those target, and similarly the SDG also we are not able to, so that the national biodiversity strategy for action plan, which is incorporated the strategic plan management plan for the invasive alien species. That is where it has also been says that by 2025 the invasive alien species, the pathway has to be identified. This is most important thing. The pathway has to identify and species control and measure to be placed in to prevent the introduction of establishment. That is one of the reasons that the National Mission for Himalayan Studies has given this large project, where we have been assigned to give the pathway of the different invasive, different alien species, and also to classify how many invasive species are present there and what the invasive load of each of the state and each of the Himalayan state. That was the main motto, and so that we have worked in different groups as in work which I will describe. And when we talk about the pathway, this is very easy. In pathway, this is the means and measures by or the different routes by which the invasive species are introduced into a new environment. And when we talk about the new environment, this may be a transport, this may be man-made, or this may be natural, or this may be intentionally, or maybe unintentionally. There are different kinds and different ways of introduction of the this invasive species are gone, and maybe a transport when it established, when it introduced to a particular species, a particular ecosystem or a particular region, it is got established, and once it gets established, it gets into the ecosystems nicely, and also once it established completely, then it starts spreading, and once it reproduces, and once get acclimatized, it get reproduced. And that's the native species, alien species, established alien species, and invasive. When alien species become invasive, this is go hand to hand, and unless we do not observe very neatly, and this is never happens overnight. That's why the observations or the kind of the invasions takes place in ecosystems, it usually neglected. And many stories are like the Parthenium, Lantana camera, Ageratum. All these species are invaded into the systems very silently and steadily. And this is the different kind of mechanism of introductions. I didn't want to go to that because it may be release, escape, and contamination provide a different kind of the contamination or the different kind of the vectors of the spread. This is the other part of the story. And as I told you, the project was given, and many people have been worked on that. And for the Jammu, Kashmir and Ladakh, Professor Rishi was there, and for Sikkim, Dr. Balbabu Chaudhary, and for Arunachal and Manipur and Mizoram, I was working, and for Himachal Pradesh, Professor Deji and Harvinder Singh were there, and Manipur and Nagaland, Professor S.B. Singha was working. And the entire report already been submitted to the ministry, and also it is available in the NMNS website. When we talk about the most important thing, that when we talk about the invasive species, any species which comes out from any other regions is not invasive immediately, because there is a different type of the stages on which these species can be declared as invasive. And the stage 1, stage 2, stage 3, or the stage 3, in stage 3B, this is a invasive, this is become established and also widespread. And all these stages can be defined on the type of the frequency of the stage of the frequency on which these species can be located, and the species can be occurred in a particular region. Because in stage 1, the potential alien species of the propagules are limited in the native ranges, and these doesn't make any differences to the native species. On the stage 2, potentially they are propagules of the transporter vectors, and the stage 3, we release and introduce to the non-native ranges to the new ecosystems. And when the stage 3, from the stage 3, the FP, that is frequency of the less than 10%, and also from the stage 3, it reproduces and establishes to the population in the non-native range. These are the theoretical things, but for declaring any of the species, the frequency has to be more than the 50%, and also the cover percentage is more than the 50%. Then only the alien species become the invasive, and we can say that these are invasive. When we talk about the invasive, this is a well-established species which affects our native ranges. While already we have also marked some of the new invasive species. Those are new invasive species are those, though these species are the more potential and newly introduced to the system, and also potentially they are destroyed to the ecosystems. And when we talk about the management strategy of these systems, when these predictions, eradication, and prioritizing of the control, and the public policy and mass awareness in all the 4 stages of this already been incorporated in the integrated management plan for the invasive species of our national level, and where I am also a member on that. There, the early detection and rapid responses is on the stage 1 and 2 and 3, and the prevention when we talk about the risk analysis and also the early warning systems in the stage number 3 and 4, and also prioritizing of the control. This comes under the stage 4, 5 and 4 and 4, 5. And public policy awareness, informed decisions, because not only the eradications and the destruction of the degraded ecosystems is important, but also this is very much important for giving creating a general awareness of the local stakeholders, because sometimes many people do not know what the species is spreading into their own ecosystem or own native ranges. That's why the creating of general awareness is one of the most foremost things to do that. And this is for the methodology we have adopted here. The native ranges we have taken from the POE, from these relevant sources, and also present study, also World Geographic schemes for the species. We have native and indigenous bromate 2001 we followed up. And for purpose of the introduction of present study and how it has been introduced, and the scientific study, the Dr. Kuru is here somewhere. And we have adopted Dr. Kuru's one of the paper from the 2007, and also different literatures what we have done. And one of my students working in the floor, this is invasion on the Mizoram, and also given the entire Tripura and also Mizoram area given. And the collection trips has been made from the 2018 to 2021, and life history traits are differently described. Along with these all the alien species, we have also called the native ranges we have collected from different range species. And when the survey sampling has been done, this is randomly located sample points have been taken in the different ecosystem, protected areas, unprotected areas, invaded areas, and non-invaded areas. And sample points along with the roadside and trailing, because roadside is one of the most severely affected by the invasion of the plant species. And gradually, this is the first point from where the plant goes into the native ranges of the forest ecosystem. And the potential samples occur every 300 meters. We have taken the two, particularly today I'll talk about the case study in particular in Mizoram, where the Famkui National Park and the Mondale National Park has been investigated, where then in 300 meters we have taken the sample points for that. And the stratified random sample points where the strata is defined for the elevation, because each of the 300 of elevation, we found that there is change of the floristic components, and how these species has been affected, this area has been studied for detail. And the habitat, the natural, artificial, both the things, as I told you, the protected areas, the invaded area and non-invaded area has been taken out. And the habitat features, we have taken whether these open areas or this is shady area, or the distinct feature, whether the told you the native species as well as the invaded species, or as well as the alien species has been collected, and all the passport data of each of the species has been recorded. And this is some of the areas where students are working. For the phytosociological analysis, we have taken out everything, frequency, density, and the relative abundance of those areas. And along that, the morphological trait and the reproductive potential of that of each of the species has been taken out. And for this, the length of the root, the primary roots, the nodal points, and the length of the internode in the different seasons. Because in the what is the impact of the different seasons on that? That also we have been calculated. And length of the shoot and there's a number of leaves, the primary leaves also and primary buds, so that we can able to know that what are the morphological traits are helping them out to spread out. Because it very rapidly the species spread into the jungle. And similarly, the reproductive trait, we have taken the vegetative regeneration, how far the vegetative regeneration, because usually what happens, the forest department, they cut and throw in the same place only. Because whenever the forest department wants to clear that area, they cut and throw in the same space. So that these areas has been invaded again by the vegetative propagator, highly vegetative propagator. So that's why whenever it has to be cut, it has to be taken out from the area. And similarly, the number of capsula on the plants, number of florets, and how the seed germination capacity of each of the species has been calculated. And these are some of the species, the alien species from located on a different location of the northeast states. This is a Problema, Tithonia diversifolia. This is regarded as the new invasive species because the frequency of this particular species is more. And Mikuna, that is also the flowering in the Manipur. This is one of the species which is spread over the different areas of Manipur. And Azuratina, and similarly Imperata, again in different parts of the Nagaland. This is from the Nagaland. And Gallinsua piflora and Lantana camara. This is though Lantana camara is not very much in the Mizoram and Manipur. This is rampantly in the Nagaland. And this is Alternanthera. This is one of the most invaded species in the northeast. And Alternanthera also again Asclepias. These are some of the species which are during the study. Mycenia micrantha, this is one of the disastrous species which is spread throughout the northeast. And while estimating the different kind of the invasive load, while doing that, we in the Sikkim, Himalayan and West Bengal, we have taken the 3 districts of the Sikkim, where the total invasive load that we talk about, the 333 species has been documented, in the Mizoram 163 and the Tripura 186. In Tripura and Mizoram, we have taken in 11 districts of Mizoram and 6 districts of Tripura. And Arunachal, Manipur, Nagaland, where we have taken 131 and 3 districts in Arunachal and 3 districts in Nagaland, and also 9 districts in the Manipur. And the invasive load, while talk about the Sikkim, again, this is the top families, Fabaceae, 58 species, that is Asteraceae, this is followed by 47, Solanaceae. These are the alien species which is found in there, and this gives the details data of what kind of, and based upon the IVI and based upon the frequency, we have designated the first 10 species which is more invasive on that, and also the more neo-invasive onto that. In Sikkim, based upon the IVI, we found that Azuratina adenophora, Lantana camara, Mycenia micrantha, and Chromolema edurata is the most invasive. And this is the characterization of the species. I will not go deep into that. Similarly, in Manipur, Nagaland and Arunachal, 131 species has been recorded. And out of the most open forest, this is the Mycenia micrantha, Azuratin conijorides, and Chromolema odorata is recorded the pure patches of in the mixed populations. This is the characterization completely how many species are Arunachal, Manipur, Nagaland, and total we have given. And the mode of introduction, if you will see that, the unintentional introduction is the highest. And that is the pie chart, where the unintentional in the sense they everywhere, and each of wherever any embankment is needed in the field, they first of all they put this one, and after that cut and throw. That's why this is one of the most reason for that. And when we talk about the Mizoram 163 and Tripura is 186, and the The maximum percentage of alien species, this is from the tropical American countries, because this is also, we have seen that from where the plants has been came here, and this is Asteraceae, the most dominant family from where all the in all the northeast states, and the most dominant invasive species here is Ageratum canephora and Micrania micrantha, and Ageratum again, and Mimosa pudi and Iponia is found there. And this is the characterization of each of the species, invasive species, alien species. And when we found that the Mizoram Tripura, this is the first 10 dominant species which is found in the invasive in nature, and where the IVI is the maximum in the fields, and particularly in the protected areas and non-protected areas. And this is regarding the Manipur, Nagaland, Arunachal, and if you see that the Ageratum adenoflora and Ageratum conijoides are the most common, and it is found in almost all the space. And when we compiled all data, then we found that the Ageratum adenoflora, Ageratum conijoides, alternate, and also in number 5, the odorata is the most dominant. And if you see the history, the different time, different in a different timeline, this has been interpreted, and for this we have taken a timeline for that, and this species has been introduced in these northeast areas. And this is if you go to the Ageratum adenophorum, these species were introduced into World War II during 1940, and the adjoining is particular for them, and for making a camouflage for the persons, those who were in the war. So that's why after these species has been spotted in these areas, the oldest herbarium site was found in BSI herbarium is 88. This is around 1825. This is from somewhere from the Myanmar, and also this is from this we have a 1932 also from the Islam. The previous presence of Adenophora is a herb. This is software, and this is found from the 700 to 3,000 meter of height in Mizoram and also in Ceylon. And you can see the spread of Ageratum adenoflora in the highest peak of the Mizoram. The entire area has been covered by this. And the conijoides, this is the same way this has come, and it is believed that during the World War only the same species has been come to here. And this is limited up to the 2,500 meter in the Mizoram. And this is the fallow lands of Mizoram. This is completely invaded. And Alternathera sessilis is also found, and this is South American ornamental plant, and it is believed that this has come to India as an ornamental plant, and after that this is some part it has been also been used as the fodder and also been taken as food. But in the northeastern states, this is spread into the protected areas also. This is Videns spilosa. This is Chromolena odorata. This is one of the another species which is found in the Mizoram and an entire Indian Himalayan region also, and this also goes up to the 3,000 meter of altitude. And the species is most abundant in the 1,300 to 1,700 meter high. In the IVI is high on that, and the aggressive natural forest, where the forest has been the June cultivation or one of the species which is the first colonizer in the June cultivated areas. This is some of the adjacent regions of the Mizoram. Ipome carnea, this is another species of tropical American plant which is found in Mizoram and also Tripura. This plant causes obstruction and difficulty for proper use, and basically all the water bodies in that area has been invaded by this. And once the water body or areas invaded by this, and due to the different successional stages, these species in the summer, this species goes into the areas and encroaches the water body areas, and in the water, when in the rain season, it again comes to and decomposes, and in the gradual 2-3 years of time, this got eutrophied, and the entire area has been occupied either by encroached to the land, or the depth of the lake also been destroyed. This is Lantana camara. I need not to speak about the Lantana camara. This is the most dangerous species, the most invasive species throughout the country. But here also the one thing what we have observed that here in the Mizoram also this has gone up to the 2,300 meter of height. So that is the one of the most observations on that. And this also it doesn't invade to the intact forest, but encroaches the natural forest has disturbed. Once the forest is disturbed or somebody is cut and it's there, then once it there, then it changes the entire ecosystem of that area. This is Alantana. This is roadsides of the Mizoram forest adjacent in the Phompuy National Park. And Mycenia micrantha, this is also a species which is completely invaded, and this is also believed that during World War II only this species has brought to India for the same purpose, the camouflage in the airfields of Northeast India. And also this is one set of this is quickly and smoother other vegetation. This grabs the other vegetations and within no effort, within maybe 6 months of time, this invades the entire area and killing all the plants and the decreasing the yield of biodiversity. And also this spread into even the backyards of each of the villages. And this is the Mycenia micrantha. This is the forest in Mizoram. You can see the aerial invasive spread of the Mycenia micrantha in the Phompuy National Park. The entire forest and the entire riverbed, you can able to see that river portion, and this is spread out. And once it's spread out, you can able to see that you cannot enter into the forest also somewhere. You only from the distance only you will able to see this one. And Parthenium haustorium, though this species has not been well spread in the northeast, but this is very well spread in the northern part of the country, and it is also gradually it is grabbing to the natural ecosystems of the northeast states. And this is the area of the forest, natural forest of Tripura. This is adjacent forest. You can see the thick forest and adjacently, and if untreated, and it can go further deeper into the forest. You can see this Lantana camera and also the Parthenium. This is the invasive spread of the Azuratina, Idenophora, Chlorolena, and Mycenia micrantha in the Mizoram. In the same forest, the three species has been combinedly. This has been spread out. These are some of the Azuratina riparia. This are also we have collected. Azuratina riparia is a new invasion actually. Though this species was not been invaded before, but not reported anywhere. So we have seen that the Mizoram and Tripura, this is a new investor, and that's why we classify it as a new investor species. And this is the Phompuy National Park. And this is again in the Morlin National Park. You can see the great blue mountain hills of this is spread like this. And when we talk about the morphological traits and both the areas in the Phompuy National Park and the Morlin National Park, we have seen that each of the morphological trait, that's the length of the whole plant, the circumference of stem, a number of leaves, a number of shoots. This has have a great impact on the rainy and also the summer season. In the rainy seasons, this is proliferate like anything, and the length of each of the morphological traits is larger than the morphological trait what we found in the rainy season. That's why it shows that it is a positive relation with the monsoonal seasons. Similarly, if you see the morphological trait of Azuratina and riparia also, this is also the summer and rainy seasons. The morphological trait is and in the negative correlation in the winter seasons or the middle altitude. Because in the upper altitude, this is having a great impact on both the rain seasons and in the summer season. While in the middle altitude, this is in the winter season. This is having a negative correlated. And again, the Chlorolema odorata, we have taken on the three species for that. And the luxuriant morphology expands in the summer and rainy season. In the lower to middle, the entire area of the different altitudinal level, this is having a luxuriant growth on that. And if we go for the reproductive trait of these species, the production of the number of capsula, the plants at the maximum, the Echinophora in the middle level, while in the various odorata, in the lower level, in the second, in the sense, the 13 to 1400, the number of capsulas is more. And that probably this is one of the cause there, the the more and more disturbance area and more and more tourist influx area, these species are invading rapidly. Similarly, Ageratina, Adenopora, and Chlorolema, the seed production plant, the highest in Ageratina, and after that the Chlorolema and Ageratina riparia. The seed viability, when we tested for the seed viability, this is again in 62% to, it ranges from the 62% to 72% of the seed viability. So that it is undoubtedly up to when it is talk about the 1200 meter to 1500 meter, the seed viability is the maximum. That's why the rate of spreading of the species into the forest is also maximum. Because if you see that the forest between this particular 13 to 15, this subtropical forest, and also degraded by different kind of the maybe in the jute cultivation, or maybe the expansion of the lands or expansion of the agricultural field. That's why once it is invaded, this is the initial colonization of these species in these areas. You can see the human settlement in Sangpu village of the Phampui river. This is the area of the human settlement, and why this is the top of the PNB, and that's why whenever, and you can see the first clearance from one side. That's why these the frequency of this all the invasive species in this Sangpu river is high, and that's why once the area has been cleared for any sort of work, and this is the initial colonizer for the rainy season, and once it is colonized, it changes the soil content, or it changes due to the different kinds of allelopathic chemicals, so that the natural forest is gradually destroying. Either the natural forest, and whenever the forest is coming up, it comes with the monoculture, maybe members of Melanostomaceae or members of Cervia, or something, the complete monoculture which is not suitable for anything for the and it changes the demography of the forest composition of the particular area. And again, this is the Himalayan Moorland National Park we have taken in 2021. You can see the human settlement in the down, that is a yellow one, and the red one in the top of the Moorland National Park, what is known as the Blue Mountain, and this is the last point of the human settlement for before going for the Moorland National Park, and we are able to see the forest clearance of the different page-wise, and those pages are the most vulnerable areas for the invasion, and because the species have already been invaded those areas, and unless not cleared or not been the local people has not been told, it will be very difficult. You can see this is the complete. This is the complete clearing of a forest for the June cultivations, and this is adjacent to the natural protected natural forest. And this is one of the most another thing where shit. Yeah. This is the clearance of the forest, and this is the complete clearance of the forest, June cultivation of the fallow land. And the fallow lands, if you see these colonizers are only the species of Ageratina and Chlorolema. This is again one preparing of the field for the June cultivation, and these are the one of the factors. These are the catalyzing factors for the spread of these species in the forest. You can see this is the entire area, and this is just adjacent to the Moorland National Park. You can see the roadside, along the roadside the forest is intact. This is aerial view actually we have taken from the drone actually, and if you see that, if you go to the nearer place, the forest is intact and very, very dense. And once this species is occur there, then again it will go to the inside of the forest. This is the fallow land of the 1 year for the Imperator cylindrica. This is again the Tithonia diversifolia in the fallow land of the PNP. Just outside of the PNP, this is the fallow land where the Tithonia is completely invaded at that area. And when we talk about the 2020, what the area has been cleared, and you can see that the initial colonization for the Ageratina species 21 and 23, and you can see able to see that the species is not given any scope for the development of native species, but it is completely occupied by it. So that's why this is one of the great concern, a great challenge for the conservation of the species. That's why whenever we are talking about the control and management of plant invasions, we need to have a prevention and the early detections and control and management. And we should have a legislation and the education. And the down three parts in control and management and legislation and education and outreach, that is a different story. And when we talk about the prevention and early detection, till now though we could not able to have detection because a stronger legislation has need for that. And if it, for that purpose, we also prepared some of the protocols for the predictions of IAAT. This is a different invasive alien species. We have taken the variables from the bioclean models, and also this is for the Parthenium actually. This is done by the doctor. This slide is from Dr. Resi from Kashmir University, and this is the Anthenium cornula. Also, this is also the predicted expanded the suitable range of the Indian Himalayan region on a predicted feature. Because earlier, at the present stage, current scenario, the larger one is the current scenario where the species has been inverted in different places, and in RCP 2.6 and RCP 2 4.5 and RCP 2 in the 2040. 2050 and 2070, this could be the prediction of the different conditions for this particular species. And also this is Coneza canadensis. This is the current scenario where the species is presently spread out throughout the Himalaya, and different in the same condition 240 and 2050 and 2070, the different RCP, the species can be spread over the different part of the Himalaya, inter-Himalaya. And similarly, we have given Hyptis, and also we have Chronorama odorata for the Mizoram. When the Mizoram, the entire point, the white point, what we have taken the samples from those areas, and the black ones are the current scenario stage. This is the current scenario of the spreading of species. And in RCP 2050 and 4.5 and 8.5, the entire area once the red areas are the vulnerable area where the species is going to spread in that. And this is a space, the predicting modeling, only the interpretation is the annual temperatures we have taken, and the annual precipitation, and because we know that the annual temperature of this area is gradually in the last 100 years it has been for 1.5 to 2.3 degree higher. So it is also expected that in the next 100 years it is also expected around 1.5 to 2.5 degree will increase. So that's why different biological biological conditions, the species of the Chronorama spread into the entire area unless it has been conquered or unless it's been not been eradicated or been taken as of that. So that's why this is if you see the global distribution of these particular three species, the entirely not only in India, this is entirely making a mix for the entire world. And accordingly, if you see that if you see India also Western Ghats, Eastern Ghats, the entire Himalayan area and the Northeast area, these species has also been spread out. And similarly, the prediction for these three species, if you make a prediction modeling for this one also, we have given in the report everything for the next 100 years. What could be the scenario of these particular three species if not been controlled? This we have given, and this is the 2050 and 2070 and different area and different bioclinical models. And we have also studied the allopatry impact on the crop plants, because the germination of the crop plants is negatively being impacted by and correlated by this particular three species, where the impacted inhibition of the germination percentage were Ageratina adenophora, Ageratina riparia. This is Oryza. We have tested for the Oryza and Lens and the Sisal and Brassica, because these are the four crops which are usually done in these tribal areas, and the forest, the entire forest, the croplands is surrounded by this particular three species. That's why we have we wanted to show that whether it has an inhibition impact or not, and we found that this is positively correlated, and Chronorama odorata is the maximum impact on the Oryza and in the Lens and Brassica and Sisal. And similarly, we also the plumule growth of the croplands, that also we found that we have taken all the seeds from the same area and the same varieties, and also we tested on the control condition and also different conditions for this mixture of the Chronorama and also Ageratina agitated adenocarcinoma, and we found that there is a drastic changes on the when the concentration increase, the drastic change on that. So that's why if the percentage of these invasive species is increasing to 50% or more, there is a chance of the change of the yield of the crops in those areas. This is again for this radical growth of the crop plants. This is all are negatively correlated. And this is also what we did actually. This because if you will only say that these species are not utilized, that we have also tested for some of the socio-economic impact or utilization pattern of these invasive species. So then we have taken 104 alien plants which has been used by the public actually for the different purposes, and this may be the roots or fruits or the tender shoots, maybe it is eaten, or maybe some of the flower has been eaten. So that's why if you see that the whole plant is 12%, roots is 23%, leaf is 39%, and fruits are 9%. These plants have been used by the different purposes, and when we taken that, because the main use for this is if you will see that latex, smokes, or raw materials, somebody is taking out oils and juices, pollutants, and out of the 104 species we have been calculated on that, and we saw that it is the fodder and also the vegetable is the maximum. This is the use at the maximum. There is a fodder and some of the species also been used for the thatching, and although negligible, this is the some way, one way or other way the people are using these plants actually. That's why whenever we talk about the bioprospection of any of the species, so we need to go to each of the integrities of the species, so that in what way the socio-economic condition of these people can be achieved or to increase that can be calculated. And the use value also we have taken from the use value and the information consent factor. From there we came to know that the respiratory diseases, people are used for the respiratory diseases. Suppose something is they have a feeling in the cold and cough, they are smoking, they mix in the smoking, the digestive system disorders, inflammations and tumors, and microbial infections and the pain exhibits in the highest ICF factor. That is what we call the informant consent factor. And this is when taken from the different group of the people, from child, women group is different, young women is different, old ladies are different, because this is an integrated report what we submit to the ministry. And this is, you can see, this is also some of the species which is found in the market, and this is sold in the market of the Phampu National Park and the Mulan National Park areas, and this is the general market in the Mizoram. And these are the species and commonly used in different purposes. And this is also value addition in the livelihood generation. This is one of our fellow is he has used this in the vermicomposting, and it has been told that in vermicomposting this species has been digested by this, and the nature of the vermicompost has also been used properly. He has used Agitator conigeries, Haustorium, Chlorolana, and also Lantana camera, and the leaves have been crushed and used in composting. This has also been evaluated through. So that for the control and management investment in Northeast, this is the most important factor what we have taken for education and outreach programs, and we have made some outreach programs. Students, the local people has been enriched the dangerous or what is the problem with this species. And we also made some Mizoram University where 2-3 outreach programs and workshops has been conducted. And you can see Dr. Sina was there at that time with me, and he is no more. And in the first department we arranged with the universities, and this is the same thing. And Dr. Watson also come, and because they are working for a counterpart Nepal, so we are working in the Mizoram. So once we invited him for give a lecture, so that in what way we can correlate our work with their work. So what we feel that what the DEFRA funding facilities what they are working in Bhutan, and they are working. They are also using our invasive species in the fodder and also in the manures. So that's why the kind of the work we also work in here in Indian counterpart is very much similar to what the DEFRA funded projects of the RBG is going on in the Bhutan and also in Nepal. And the summary, I can say that the increased global transportation, there is a continuous increase of there is a continuous increase of spreading of invasive species in the north area, and this is a very unfortunately this species, what are the species has been going into, because day-to-day life we always see to the forest area. We are not able to observe in what rate and what kind of, in what areas it has been spreaded, but it is gradually and very silent killer way this is going up, and this needs a creating of a general awareness and a general great awareness, and also enrichment for this outreach program for this general what we call as the local stakeholders, particularly the local inhabitants, and from the child they must know that this plant is good and this plant is bad, so that then only the area or the ecosystem can be survived. And the way forward, we can say that mitigation climate change and mitigation climate change and invasive species protection. As we look forward to as a commitment for the nation's commitment towards this, all the you may say the national invasive policies or the age models, post Cumming Montreal Protocol, post global rights framework. We must work along with together. Then only it can be possible. Thank you very much, sir.
Thank you very much, sir. And this was a very pertinent topic on invasive species. In fact, I'll just to sell my department here. Our Department of Ecology and Environmental Science, Assam University, is also working on invasive species, and Dr. Panadip, one of our colleagues, is engaged in it. I guess we could do a collaborative work on that too, sir. And also I want to tell you that one of his senior predecessors, I won't name him, always praises him a lot. He's a very sincere, hardworking and dedicated scientist of India. So a good applause for that. And it is coming from a good place, so we can believe in that. So sir, I will request our session chairperson and our session co-chairperson to please come and felicitate Dr. Das. Thank you, sir. Thank you so much. Now, at the end of this technical session, I request our organizing secretary, Dr. Devjyoti Bhattacharya, to kindly felicitate our session chairperson, Professor A.P. Das, and our session co-chair, Dr. Tajuddin Khan, for their patient hearing. Okay, so with that we come to the end of technical session 8 of A2, and I also, with that, I take leave. I thank Dr. Devjyoti for giving this opportunity to be with all of you, and we will continue with the session after tea. Tea will be served here itself? Okay, so tea will be served here itself, so I hand it over to everyone.
one lead lecturers to avoid competition. So it is my privilege to invite our esteemed person or judges, Dr. N. Sanjappa, Dr. Pandavana, Sir Gigi Mehti on this stage. Yes, sir.
Can we start?
Sorry.
Good afternoon, distinguished delegates. In this session, technical session 9, we have a lead lecture by Professor Dr. G. Pandan, and followed by that we have Professor A.K. Pandey Award for Revisionary Studies. Then followed by that we have Professor Santosh Nampi, IAAT Young Scientist Award. So all the Dr. is already there, and the other award lectures should be ready, and all one by one.
Okay, thank you, Chairman.
Please go ahead.
We are already 1.5 hours behind the schedule, actually.
That's why I'm not introducing you, because all of them know you.
Yeah, I'll try to complete it within 20-25 minutes. So quickly, the purpose of this my talk is actually for youngsters. Actually, how Convention on Biological Diversity, in fact, gave a new lease of life for the taxonomies. Actually, I just want to introduce a few slides on CBD and taxonomy. Some slides are meant for youngsters, so he has to excuse me. Right now, when the CBD came into being in 1990, but the years of me, many of the youngsters will be almost a few years old. Actually, they will not be aware of the but in the process of learning. So I want to know, just take you a few slides, what CBD says actually. CBD is, as I told you, it is an outcome of 1992 Rio de Janeiro conference, and there were actually three conventions were raised. Actually, one is CBD, second is a UN Convention on Climate Change and Certification. There were two, but since biology is more we concerned about the Earth Summit, particularly the CBD, what it says actually, conserve the biological diversity for posterity, use biological resources sustainably. The word sustainability get prominent from then onwards, and then share the benefits fairly and equitably using the genetic resources actually. These are the three pillars based on which the CBD stands actually. So this is the framework. What is the CBD speciality? This is a global framework for action, to devise action for conserving the biodiversity as a member countries, UN member countries, because this is actually under the auspices of the United Nations actually. So at the time, immediately, there were the decisions at the national level. Recognition has to be as a sovereign properties of the every country, where particularly, and by using this benefit sharing, the benefit sharing will be through funds or the opportunities and technologies. So at the time when CBD to be 187 countries, but till Nagoya Protocol were that United States was not the member of this party. But when the benefit sharing mechanism was introduced, and they are the first country promulgating the what you call the act for benefit sharing mechanism, they become the party. Now, right now, about 198 nations. That means 197 independent countries and one entire Europe Union actually part of it. See, the first time, though we were aware of the genetic diversity and all, but the framework was formed for what is that speciality is the biological diversities within the species. We are recognizing within the species, between the species and ecosystems and landscapes, so that three levels were clearly they are demarcated and then identified, and then started valuing this biological diversity in a aspects. Till then it was actually an academic, we were looking into that. The importance of the biodiversity, the material services we are receiving from them, ecosystem services, recreational and economic potential. When you come into the what you estimate in a financial value, I just want to spread there, tell you, these are annually by farmers, botanical science, agriculture, ornamental, crop protection, biotech and personal care. The minimum will be 500 billion USA dollar per year to maximum 800 billion dollars derived from the materials received from this biodiversity. So this is what I just want to, from that point onwards, people started converting the biodiversity into economic aspect by using the word tangible and intangible. This is the word was very familiar from 1992 on. See that we know more than 7,000 species are cultivated or collected for as a food crops, but the entire world is fed by only 30 species actually, of which 3 species particularly provide 50% of energy intake for the human being, particularly paddy, wheat and maize. These are the 3 crops. Medicines about 25 to 50% are still derived from the plants, and future medicinal properties to be explored to safeguard what we call the security of our nation, every country, particularly on the medicinal aspects, health aspects. See, habitat threat, you know what are the threats to the biodiversity? So habitat loss, just the previous lecture, invasive species, biology and value itself is actually one of the reasons for a species to push into the extinction mode, pollution and climate change. And so now I just want to impress what the impact after CBD came in. This is really a positive impact or negative. This is what when we were talking about taxonomy and taxonomy's importance and their what do you call the taxonomies are extinct, but right now I am giving you the actual details from the IUCN and the World Conservation Monitoring Center data I have collected. During that period, that is 1992, when this CBD was promulgated, it is 30,827 plants, 5,200 animals were in threat categories, and microbes were not known. But how the CBD operates, CBD when they are operating, when we are telling about the 3
biological, that is the pillars on which it operates. But there is a lot of what you call the conflict of interest when globally trying to implement. So the CBD Secretariat has developed several subsidiary bodies to overcome or to compromise or to settle the issues at but national and global levels. These are the different mechanisms actually being operated. And if you see the thematic work programs where this CBD is actually being implemented, that is agricultural biodiversity, biodiversity of inland water, biodiversity of dry and sub-humid lands, forest biodiversity, marine and coastal, mountain and then highland biodiversity. These are the thematic programs. Every member country has undertake based on that these programs are being implemented. But when we are started implementing these program, there will be cross-cutting issues are there. When you go for traditional knowledge, how to protect the traditional knowledge, then they try to develop